Everyone, welcome back to lectures 14 and 15 on protein and amino acid metabolism. This video here will specifically focus on some of the key reactions of amino acid breakdown and interconversion. And this is the second part of our video series. So if you haven't watched the first introductory part where we looked at the general overview of the utilization and interconversion of proteins and amino acids yet, I would encourage you to do that first before we're diving in to the more specific enzyme reactions in this and the next video. So as for our learning goals, right, we had started in the first video to look at the general principles and concepts of how proteins and amino acids are used in plants and animals. And we had started to look at the different metabolic fates of carbon and nitrogen in amino acid breakdown reactions. I want to build on this now and specifically cover the material that will allow us to describe and eventually apply the reactions of these aminotransferases that are really key in amino acid and to some degree also carbohydrate metabolism. And in this context, we also have to understand one of our key cofactors here, PLP. So watch out for this other video, the supplemental video, where I'm covering the two cofactors that we will encounter as part of our lectures 14 and 15. So to summarize again from sort of the end of the video part 1, the introductory video, we had talked about this overall pathway on how we can utilize dietary or internal proteins, how we can break them down into peptides and free amino acids using hydrolases, and how we then can use all these different amino acids in these aminotransferase reactions here to bring them to glutamate as sort of the central amino acid in the catabolism of proteins. And then glutamate, we had talked about how it can be deaminated to release our ammonium, and that can either be funneled through resynthesized other amino acids or primarily would be removed, excreted through urea for us as mammals, right? So within this overall pathway that we had discussed, this video will really focus now on these critical aminotransferase or AT reaction in the liver cytosol that allows us to interconvert amino acids using alpha keto acids here as intermediates. Right? These are fully reversible reactions. There are numerous of those, and they're absolutely critical, not just in this breakdown pathway here, but also for the general interconversion of amino acids. So how do these reactions work in general? Right? So we have two substrates and two products just to orient ourselves. And I try to color code the reaction here to illustrate a bit better what is happening. So say you have broken down a protein, right, into a number of amino acids. Now you want to further catabolize these amino acids. This could be any of your 21 amino acids, essentially. There are a few exceptions, but we'll just assume this for the moment. So this is your first substrate, and just point out here, right, the amino group of your amino acid. And your second substrate is actually an alpha keto acid. So again, right, think about your carbons. Right here you have your functional group. It's an acid, so it's a carboxyl group. Your alpha carbon is right here, right? And you have a ketone function added. So if we're going back to our rules of decarboxylation in that particular case, and we talked, for example, about our PDH and TCA cycle reactions, this is the same kind of acid. It's an alpha keto acid. We're not doing decarboxylations today, right? We're doing interconversions, but it's a keto acid. And so if you actually compare it here, it's very similar to your amino acid with the difference that this one has an amino group at the alpha carbon and this one has a carbonyl group at the alpha carbon. What we essentially want to do is to switch them over. And that's what the aminotransferases in essence are doing, okay? So let's have a look here, and we can use alpha Kg and glutamate as examples because in the most reactions, especially when we're talking about amino acid breakdown, alpha Kg will be the universal amino acceptor. So it will be the most common metabolite to accept the amino group. And glutamate will be the universal amino donor, right? Like we had shown in the overview, everything culminates in glutamate to give over the amino group. So in these reversible reactions, those are two major players. So let's assume we're working with alpha Kg and glutamate. So you're bringing in your desired amino acid that you want to break down, And your second substrate for the aminotransferase is your alpha keto acid here in blue. What is happening actually is that the amino group here will be cleaved off and will be attached to alpha Kg, your alpha keto acid, will generate glutamate, right? Here's your backbone of alpha Kg. Here in red, the amino group of your incoming amino acid. You're generating a new amino acid, and this is glutamate if you're coming from alpha Kg. The second product you're releasing now is your original amino acid, now not carrying the amino group anymore that has been transferred. Instead, it's now carrying the carbonyl group from your incoming alpha keto acid. So in essence, what we're doing here is we're switching the amino group for the carbonyl group. So a mutual exchange of these functional groups. And this reaction is fully reversible. There's an important cofactor that is actually important here. That's PLP, or pyridoxal phosphate, or vitamin B6. So look at the additional video there where I talk a bit more about what it actually is and why it's important. I won't go into the detailed mechanism of PLP. It will be sufficient for us here to say that PLP actually is a cofactor that holds the amino group in place after it's being removed from the amino acid. It will actually be held in place by PLP. Then the protein will take in its second substrate, the keto acid, and PLP will give over the amino group. So it's a critical cofactor by simply keeping the amino group in place until the entire reaction has concluded. So it's a two-step reaction. And I know it's somewhat complicated, so I'm trying to break it down even a bit further. So you can think about it as a step 1 and step 2 reaction. Right? Your first reaction would be your amino acid is entering the enzyme. PLP is bound to the enzyme. And the first part of the reaction is that now you're cleaving off the amino group. The rest of your molecule is your keto acid that will be released from the enzyme, and the amino group will stay behind in the enzyme bound to PLP. After the keto acid, as one of the products has left the active site, the second step can start, right? You still have PLP with the amino group bound. You're bringing in the second substrate of this reaction. Now it's your alpha keto acid, your alpha Kg. PLP will hand over the amino group to the alpha keto acid. You're releasing a new amino acid, and PLP is ready again for a new reaction. So it's a two-step reaction with two substrates. So here's another way of maybe explaining it, trying to animate it a little bit so that everybody can understand what is happening in these two independent steps. This is step 1. You're bringing in your first amino acid. PLP is bound to the enzyme here. So the amino acid is coming in, and amino group is cleaved, and your first keto acid is released. Right? The ammonia group stays behind now, bound to NS3. The active site is open and is ready to accept now the substrate for the second step of the reaction. So now the second keto acid is coming in, is taking the ammonia group from PLP, the condensation occurs, and you're releasing a new amino acid. Right? So a two-step reaction in one enzyme using two substrates. And this can occur with almost all of the amino acids, and you can use them to interconvert amino acids with each other, or specifically in our breakdown to generate glutamate from all these different amino acids for deamination and removal of ammonia. So to give you one example, outside of this for another amino acid here, so for example, right, again, we could use our glutamate here, our universal ammonium group donor, and we could react it with an alpha keto acid, in this case, our OAA, oxaloacetate, right, our intermediate of the TCA cycle. We're running the aminotransferase now, right? What we're doing is we're transferring over the amino group of glutamate, and we're switching it over for the carbonyl group here of OAA. We're generating a new amino acid here. So this is a carbon backbone of OAA, now with amino group from glutamate attached, that's aspartate. So the corresponding amino acid of OAA is aspartate, and the corresponding alpha keto acid of aspartate is OAA. What we're releasing, right? Removal of the amino group of glutamate and taking over the carbonyl function from OAA generates a corresponding alpha keto acid of glutamate, which is alpha Kg. So it's just, again, one example on how we can use this, right? And it could be used in this direction, for example, right? You have aspartate, you want to break it down, you run it in this direction, you generate glutamate for further deamination, but it's reversible. Again, you can also run it in the other direction. Say you want to make aspartate. Now you would take OAA from your TCA cycle. In this case, your TCA cycle functions as a linear pathway with the purpose of generating precursors for biosynthetic pathways. You want to make aspartate. You're using glutamate in this case, and you can generate aspartate, and you're releasing alpha Kg. So you can refill the TCA cycle essentially at the stage of alpha Kg. That can run through the TCA cycle to refill OAA. So it's nice and tight, right? And you can use it in both directions for different purposes. This also should look familiar from some of our shuttles, and I'll get to that in a minute. Right? So in general, right, aminotransferases all catalyze the same reaction. This reaction mechanism is conserved among all aminotransferases. What they differ in is in their substrate specificity. So we have a whole number of these aminotransferases. They're typically specific to a specific amino acid that they convert. So they would be called, for example, an alanine aminotransferase that's specific to alanine, or a histidine aminotransferase that would be specific to histidine. So these reactions are critical not only in amino acid breakdown, that's our focus today, but also in amino acid synthesis. Right? If we, for example, like I showed you, want to make aspartate from glutamate, we can use these AT reactions. So they're a critical part of the general interconversion of amino acids, and also in some of these metabolic shuttles. So let's get to that. How does this work? It actually looks exactly the same. This is part of a shuttle I just showed you. Right, so remember, we're using here OAA with glutamate. We're running our AT reaction to aspartate and alpha Kg. And this is the exact reaction we have right here. Right? And so you might remember the lower part of the shuttle here. This is your malate shuttle. Right? We had talked about the malate shuttle already when we talked about fatty acid synthesis, our GNG, as well as our ETC for the purpose of bringing in NADH from glycolysis to the ETC. And the purpose of the malate shuttle was, right, to transfer the electrons from NADH over the membrane of the mitochondria into the cytosol, or vice versa. Right? So we had used malate, and we had used the oxidation to OAA to generate NADH here, and vice versa. We could do it here too. So exchanging the electrons from NADH without actually bringing a cofactor backbone across the membrane. But at the time, what I had told you, right, is not to sort of focus on the other part of the shuttle here, which is this one. And so now these aminotransferase reactions are coming in, because the malate shuttle is not complete just with this reaction, right? Because you're leaving the OAA backbone in one compartment. Right? If you run the shuttle, for example, in this direction, then you're generating OAA, but you have taken malate out of the cytosol. Right? In order to be a full shuttle, you want to cycle this to make sure you're not depleting carbon resources between the compartments. And so what you're using here is your AT reaction. You're converting OAA, the keto acid, into its corresponding amino acid, aspartate. You're using glutamate as the ammonium donor. And that's a really good reaction because it's simple and because it's fully reversible. It actually doesn't require any additional energy, right? It's ATP independent, for example. And now we have transporters for all these metabolites. There's transporters for aspartate, for alpha Kg and glutamate. So each of those can be transported efficiently between the membranes. But now doing this, right, if you have used your AT reaction, you can transport aspartate back to the cytosol. You can do the same AT reaction. It's simply an isozyme of this very aminotransferase. This one just happens to operate in the cytosol. And now you can do the reverse aminotransferase reaction, converting aspartate into its corresponding alpha keto acid, OAA, you're regenerating glutamate. So this now can flux continuously without any loss, you can transfer our electrons from the carrier completely between the mitochondria and the cytosol using these two reactions, the reduction-oxidation reaction between OAA and malate and the aspartate-OAA aminotransferase reaction. So a very nice and tight shuttle. So this concludes sort of our section on the aminotransferases. This is really critical reaction for interconverting amino acids. So I would recommend that you sort of go over it a couple of times to really understand how these two steps of the reaction work. And we'll apply it later on when we talk about the breakdown of a few examples of amino acids again, so we can recollect some of these concepts then when we speak about some of these specific examples.